Okay, we're going to start the report back session for this last breakout session. I have to say, I mean, I've heard from other groups. I know in my group that the conversation was actually really incredible, and I think there was so much that came out of it that we're definitely going to be trying to find some way to capture it in some form, but certainly there were scribes, as I said before, taking notes and so forth, so we'll do as best as we can. What we're going to do here, however, we're actually trying to get everybody out earlier than scheduled. The reason is that there's some bad weather coming through later in the evening is what I understand, and so we're going to try to get people through dinner and so forth at a leisurely pace, but you know, not too leisurely. So what we're going to try to do is wrap this up by 6:30, and then we'll have instructions. Christoph will come up, wrap up the day, talk about dinner, which is going to be amazing, and get people instructions for getting to the venue. So what I'm going to ask the moderators of the sessions to do is just hit the key points in 4 or 5 minutes. We're not going to take questions. I would encourage you to continue the conversation over dinner if you're so inclined. But if that makes sense to everybody, we'll try to get everybody out of here by about 6:35 and end the session by then. Okay, so I need to remind myself. So the first group one was gut, pancreas, and oral craniofacial, and that would be Nir moderating that. Yes. Oh, Nir, sorry.
Do you want to run the microphone? Yeah. Is there on the microphone too?
That's okay.
Okay, so no slides, we can do it quickly. So I was moderating the session of a biological network session number 1, gut, pancreas, and oral and craniofacial networks. We started with an update from all the networks. It seems like there is a lot of activity going on. Hundreds of thousands of cells were mentioned in all cases, multiple samples, a lot of activity, and some of the networks, the pancreas, for example, we're talking about starting to assemble a large collection of data sets and putting them together into an integrated atlas, and also starting to look at spatial transcriptomics and multimodal analysis as their next step. Now, in terms of the subjects that we covered, we had a lot of conversation. We talked about the annotation efforts. We talked about the sufficiency of the data to actually define an atlas, whether people feel like that there is enough information, diversity, which I'm not going to cover here, and metadata. There was a lot of discussion on metadata, and of course, computation underlined all of the discussion. So just to give some vignettes from what we've seen. So in terms of the annotation efforts, one of the questions that was very practical that I asked people is how do they stratify their samples? Are they doing it per tissue, for example, or per area of the organ, or per compartment? And there the consensus was that the work is per compartment. That's an easier way to work and an easier way to annotate things. And that's not always the first choice, but that was interesting to hear. There was a sense of thinking about the integration problem. So the integration, and there was a consensus about that, it should only happen up to a certain level. So when we start to integrate many samples that might be separate from each other, we can get one level of resolution of cell type annotation, but that can be much crisper and much higher resolution when we focus on very specific subset of samples that can come from either a single donor or from a group of donors that have a similar background or similar features that make them more integratable together and less losing data due to what we perceive as batch effects, but can actually come from covariates from the data. And maybe one last point that I want to make is about metadata. So there was a lot of discussion there on metadata, and the main complaint was about the need to access metadata that can be very important to analyzing samples. If you talk about the gut, right, so it's about diet, clinical history, and other things that can really affect the information that we are seeing. And there was one thing that also came out from this discussion that was very nice. It's about the need to actually integrate and bring together different networks that are looking at different organs, but all of them are basically basically had to do with the digestive system, basically, if you will, from the mouth to the gut, and bringing all of this together, because of course in the body we don't have these artificial layering between these organs. So that was also a very nice initiative, and hopefully this will catch up. So thank you very much.
So the next group is group 2, which was liver, lung, breast, and reproduction, and that was moderated by Donna Pear.
Also had a lively discussion with a lively bunch of very, very different networks at different stages. Again, we kept coming back and back again and again and again. That the biggest pain point is data integration of every flavor, whether it's integrating across modalities or even integrating or especially integrating within a modality across groups, across samples, and how to do that well. We came back to that as, I think everyone was happy to see that they're not alone, and that's the biggest pain point. We also talked again about even annotations, particularly, okay, you have the cell types that are special cells for your organ, but then you have all the immune cells and the endothelial cells, which some of the domain experts aren't even the best, so everyone brought in that, it would be wonderful if the immune atlas helps, really drive across atlas thing, really taking what Sarah spearheaded with her approach, but really bringing in the immune to study immune cells and endothelial cells across our tissues. And what we had was really special because we had lung, which was most, as you saw a couple times in this talk, most furthest along in their integration challenge and efforts, and a real resource. And I'm going to bring her up to the stage because she didn't get the chance and make John angry that will be a little bit longer. She really shared her wisdom with everyone about her data integration success. And I think it's time that she gets the stage and gets about a minute to give everyone tips on how she succeeded so well.
Thank you. Yeah, Dana just asked me this 2 minutes ago, so I tried to prepare a little bit. She asked me to basically summarize best practices for atlas building in 2 minutes. So what I could think of is something that I think, looking back, was very, very rewarding for us was to really invest time in the beginning in getting the right data together. So the first thing I think that turned out to really help us well was that we had quite a lot of different data sets, which I think made integration a lot easier. That's something I learned afterwards because I wasn't so sure in the beginning if that would work. Really diversifying, let's say, the batch effects that you have in your data also enables you better to remove those. Then investing time in getting cell type annotations and harmonizing those, that also really helped the process for us. Same for metadata. Invest time in getting those and cleaning those up, and this is very tedious work, but it actually helps a lot once you start doing analysis downstream. Then one thing that I was really happy about as I was working on the computational side is work together with people who know the biology, because we often were able to think about things computationally, but not so much in terms, or it was more difficult for us to really assess, are we able to see real biology in this integration, and does it help us understand things better? Because you can only really assess that if you have the proper background knowledge. Then, yeah, take the time, I'm maybe over time already, but take the time to really assess the quality of what you've created. That takes some time, but I think it really is also important before you bring out something that you know it's actually good. And I had more that I thought of, but I can't think of it, I guess.
So again, thanks for Lisa for putting her on the spot, but really I want to reiterate that some of the success is Lisa's awesomeness, but some of the success is really that the biological lung community totally engaged with her, communicated with her, and they developed a deep, meaningful interaction to help each other succeed, and that was a critical component. And next group.
Thank you. Group 3, kidney, adipose, and immune, and Aviv.
Yes. So we actually had 3 things. When I looked at my list, I was like, could it have been any different than that? Immune probably has the most data, but it's all over the body. Then kidney is this super focused thing in one organ, and I knew it was a really well-organized effort. I used to engage with them a lot. And then adipose is in early days. And so indeed, the lessons from each of them are different, but I want to start actually with something we all put an exclamation mark around that Matthias mentioned from the experiences in how some of the work in the kidney community, not in HCA, goes, is that we need the participant's voice, he said. That's actually the people from whom we obtain samples and whose bodies we study, and we don't have that in HCA. So that is a point extremely well taken, and we're gonna take it to heart. The second thing we learned from Matthias was one of the successes in kidney is really that they know where their samples come from in an anatomical context, in actually a pretty diverse organ anatomically, not as diverse as the brain, Ed, but, you know, maybe the next cousin in that, and that they have really leveraged the legacy knowledge in anatomy and physiology, and their most senior participant is an 86-year-old scientist whom they brought out of retirement for this purpose. And as Matthias said, this knowledge is actually literally dying, and we need to bring those people into our community who have this expertise, or we will be faced with an anatomy that we don't understand sufficiently well, and where the knowledge, legacy knowledge might not be there. Now with this, I'll turn to the specific atlases. For immune, one highlight was there's immune cells everywhere, and everyone wants help from the immune network for that reason. But they have to think about the order in which they would do things, so they're going to tier things. First, primary immune tissue, where it's primarily immune cells, like, you know, your spleens or your tonsils or lymph nodes, and then mucosal barriers were second. But after the discussion, we're considering that maybe kidney comes in as well. They're so ready to go. Other particular points from the immune system, I'm not going to be able to cover everything. It was a very rich discussion, was that our obsession with categorizing cells into buckets called cell types is really bad in the immune system. It doesn't work. And so they have a challenge, a computational challenge, and a biological challenge for all of us to address. This was immediately mirrored by the adipose and kidney participants saying, we have these immune cells and we don't know how to annotate them. And we heard that that was a feature in other breakouts as well, except that we had Chloe, so we had an edge. On the metadata side, that metadata is really variable right now. It's a lesson learned that we need to identify at least minimal metadata within a bionetwork that is essential to collect, also because there are IRB constraints around some of the metadata, and so it's unrecoverable later on. And finally, to the question of disease, in immune apparently there are 2 schools of thought, and they're organized by time zones because their meetings happen in different time zones. So the Asia-Pacific contingent, including Australia and Asia, believes that disease should be included. The Europe time zone believes that healthy only, and so they compromised to include from disease, infectious diseases, vaccinated individuals, because these are actually healthy people being, you know, being sick and then being healthy as well. In the kidney, very quickly, this is a very, I would say the situation is extremely under control. Every question we asked had a very sharp and clean answer. They have an annotated anatomy, so their biopsies are really well guided. They know where the samples come from. They have multiple data sets. They're all cleanly listed. By August, they plan to do another release. They include age diversity, extensive spatial data. They are actually the benchmarking partner for STWG, the Standards and Technology Working Group. So they have they get extra from there. They have good disease connections to the Kidney Precision Medicine Initiative and other initiatives, and they even have a connection with H3 Africa for diversity. So really a poster's child, I have to say. On ontology, they worked with Cathy Berner on the ASTC+B, as kidney was the pilot for HubMap, but they do need help on immune. So at least there's something there, and they have not decided yet between, sorry, my hair. Between integrating all data together or first curating a part as a reference and mapping to it. So there is a fantastic opportunity. And then finally for adipose, This atlas is in early days. There was a big technical barrier because of the nuclei versus the cells. They've now passed it, so they have data. But there's a lot of fat depots throughout the body. Few data set as of yet, not yet harmonized, and still quite biased in terms of the fat depots that have been targeted in those, and they have a big integration challenge between the cells and the nuclei, and likely will use nuclei as their primary reference and then map onto it, because otherwise they don't have adipocytes, they just have other accessory cells. Metadata is crucial and different than what you usually think of. For example, they need metabolic parameters for the data to be interpretable, and those are not historically typically captured. So quite distinct. And in addition to primary fat depots, there's also fat cells that are embedded in other tissues, and so they come along for the ride in other data sets. They're not their first primary focus, but they will get to them eventually, just like with our immune community. And then finally, we learned really interesting things on opportunities across atlases. Collecting paired PBMCs when you collect data from your primary organ was something that came up in a conversation between the immune and the kidney people in our study, and that is an interesting connector. Both adipose and immune really need a landmark map across the entire body to know where their samples come from, and they thought that they could partner on that and try and develop this kind of information. When you collect your tissue, if there's a fat depot next door and it's accessible and available, the Adipose network would be very excited about this. And computationally, in addition to addressing these continuums, the spatial data is the challenge for kidney. We were told that having websites that are not just data, but include protocols and information about how data is being collected and the metadata that matters and so on would also be very valuable. That's it.
Excellent, thank you. Okay, next we have Fabian with session 4, which was development in nervous system, organoid, and eye.
Hey guys, so we spoke about the dope organ networks, development, organoid, brain, and eye. And sorry for that. We actually spent most of the time on understanding and sort of getting updated, but that was really fun and great discussion. We learned a lot, for example, in development, a lot of multiple data sets around integration by organ seems to be quite doable and realistic and actually sought for, something like 400 samples, nearly 10 million cells. Challenge was that in development, obviously, there's much more de novo annotation needed, and there's just gaps in the time non-homeostasis that you have that could be potentially computationally filled, maybe sort of combined with in vitro. And in the end, you end up doing, if you don't do it by organs, like if you do the bird's eye view with a mini HCA, so it's a big challenge. Brain, very exciting, very homogeneous network in the sense that sample accessibility is so non-trivial that, and sort of Alan Brain has been doing this so early on that there's been a lot of sort of very organized, high quality, also spatially, as well as function resolved data coming in. Particularly ready also for cross-species analysis, particularly also something, a lot we learned from mouse where they've been essentially, how did Ed say, they're done? And then we sort of discussed what does done mean and so on. It was quite an exciting discussion. Challenge was that n equals 3 and general sample availability is not so high, so the sort of diversity part on that is going to be a challenge. Organoid, we discussed a lot as sort of this vision of using that as a perturbation and as also a screening system. The network has been starting considerably later. I think, as Christoph said, is ramping up, and the opportunity is really to map a lot of the existing atlases that we are building in the other systems to that in vitro case, and also, how did Christoph say, to keep us real and to sort of use it also as a QC to see how well, for example, a particular protocol works and so on. So that was a very exciting. Challenge is that they are now reaching high throughput. There's a lot of data sets around already, very distributed though, and the idea would be now to leverage this diversity, but it would be challenging now to create, sort of to pick those that would be sort of HCA level quality, and this is something they're building together, and they're building this ramp up phase and sort of getting community together. I was an online presentation from Rui, who unfortunately couldn't join. He very explicitly described that they're also ready for putting things together. It's actually something that's starting. They have something like 3 million cells. A big bunch of that, 2/3 or even more, is from retina, where they have highest variability, so that seems to be making sense. ATAC as well as Murfish is about to come in. It's very comprehensive. The challenge was that at this particular session, that Rui wasn't around, and there was nobody actually from the iNetwork there, so it was not entirely, was sort of the discussion, we didn't intensify the discussion. Then we discussed what should be in Atlas v1.
Yeah, so what will be an Atlas version 1, primarily for development, braid and organoid. So we discussed about minimum amount of data, modalities across networks. The brain was more advanced with spatial, but the consensus was that we would include all cells, but only healthy samples. And the second component about the V1 Atlas for the 3 networks primarily was how diverse or inclusive should the samples be. And this is particularly difficult. Brain is hard to access. Developmental samples are difficult to work with without biobanks. And also how high throughput can we be with organoids. So I think one consideration is, should we take into account of disease disparities affecting these organs or these particular systems, which is associated with ancestry, or Fabian came up with another great suggestions of how we could overcome this, and I'll let him explain in the rest of the update.
So we thought about potential of leveraging also cross-tissue to then drive maybe a study of variability in systems that you can do. For example, in PBMC, we do have the genetic variability already happening. We can start to do GWAS and so on. So there was a discussion idea whether we could use that to test drive and then think about, let's say, imputing particular state and variability. Once you have sort of one given state and you see variability here, can you sort of impute that? That's something that computer vision does with machine learning a lot, and we could see that. And we won't believe those imputed ones, but we would use this for experimental design, for exploration of variability that could putatively there to then sort of really be informed where Ed should put the next budget on to really then get that from. So that was this discussion of sort of informed experimental design. The last point we made is, what do we now need to do to build the Atlas? And then we're done, just 3 short points. There was very clear expression from each of the 4 networks that were discussed that they are very interested in actually doing in parts already integration within the networks. Very clear wish in the discussion. There was some care to be said with respect to validation, to not overcorrect. And we spoke about the computational challenges. For example, how do you do with organoid versus development, how's the data sharing happening sufficiently early? In COVID, we did a very successful early data sharing policy, and Musk came up with a Fort Lauderdale principle from Human Genome Project to maybe sort of push that. How do we plug in, second point, into existing systems for analysis? How do we sort of use organ networks? Should we be flexible in this approach, or should we just sort of build on one standard? And I think it was quite clear in the discussion that the flexibility would be the way to go, just to have something out that the others will use. And that was, I think, one of the most exciting points at the end, namely that this integration across networks was something that is very built into particularly our organ systems, right? Because, in development, there's obviously all these organs around. In the brain, astonishingly, there seem to be very many developmental signatures, and you want to map this across. And in general, there was this fear that if we think too much in silos, we will be missing out on this part. So how do we push for that? So how do we sort of make sure that we do this? How do we add the perturbations that sort of organoids really enable to compare, then maybe also development, adult, and so on? And that last idea was maybe some internetwork video conferences, matchups, maybe at the next developmental Atlas meeting in fall to get people together along this line. Thanks.
Okay, thank you. And then last group, Malta. So the last group was heart, musculoskeletal, and skin.
Exactly. Yeah, we also had a really interesting discussion just across the road at the hotel with 3 different bionetworks at different stages, starting from kind of muscle, which is still a very early age, surveying the field of what the community, also people who are not working on single cell, would like out of an integrated atlas, and was projecting that probably in the next 2 to 3 years, there wouldn't be very many single cell data sets out there, but they're very much in the phase of community building. In heart, they're already successfully integrating single cell across single nucleus, but not so much yet across different data set batches to really build larger representations. And in skin, there was a concentrated focus group which had come together to identify 11 data sets as kind of a first pass to try to build an atlas of young postnatal up to adult healthy skin. The challenge there being that on all three, we then kind of focus on challenges that are common to different bionetworks, specifically sampling different locations. The skin is very, very large, and that's difficult to kind of build, have consistent samples, enough samples in different data sets across different regions to build one integrated atlas. Another challenge that was common was metadata aggregation. Specifically, support was asked for kind of reaching out to all the different data sets that are out there, the kind of also understanding which metadata categories would be important to harmonize and to gather. And there we kind of came to kind of solution proposals for interacting with the DCP, who was also representative present there, to identify with the individual bionetworks which categories would be important to keep, and then using some of the templates we have from the Human Lung Cell Atlas also to kind of apply this in different contexts to easily engage the community. But there was still some requests for further support for specifically contacting and this kind of community building effort that is required to get buy-in from all the different dataset generators. Regarding diversity, often it was mostly white male, not only males in the heart, both male and female data sets available, but age in the heart over 40, which was difficulty kind of getting a larger diverse representation. It was mentioned specifically that generating diverse atlas was difficult due to also high sourcing samples in Africa and high sequencing costs there, and whether one might, it would be a good idea to reach out to 10x and other companies to facilitate this. In terms of integration in the skin, which was the furthest along in that effort, there were difficulties of different sample sizes leading to different different representations depending on subsampling or not when you integrate. Also, not all metadata was accessible at this point, so making it difficult to evaluate the quality of those integrations. In muscle, only one data set was currently accessible, so that question wasn't posed yet and it's still a very early stage. In terms of annotation harmonization, we discussed as well. This was apparently not too difficult in terms of just harmonizing labels, but the difficulty was basing this on an integrated embedding where all the three bionetworks were not quite there yet. And finally, we just kind of do a quick round for why we want an integrated atlas. And after a short pause to think, it was mostly about kind of consensus annotation markers, building a baseline to which compare to disease for. And that's pretty much what we discussed.
And then last but not least, let me ask Shyam to come up and add some perspective from the genetic diversity group. And then we'll have Christoph send us off and invite us back.
Tough job coming after all these people and standing between you and escape from here. We've discussed from the genetic diversity breakout session, we've given you the general overview. So my point here is to say, what did we learn from these organ-specific bionetwork sessions, breakout sessions? Our mission here was to recruit teams who might have diverse data sets and say, can you contribute your data to the genetic diversity network? And recruit them as partners also. Can we work together on the data analysis? So we do shared data analysis and then use it for our respective purposes. I also want to clarify to anyone who might be thinking of contributing data for this effort that the idea is not to replace whatever you're doing in your network or to ask the same scientific questions you are asking. We, of course, make sure not to do that. Rather, the idea is to do cross-cutting analysis of a few basic traits, a few basic axes of human variation across many tissues or one tissue across many teams. And hopefully, you'll all benefit from that. You'll get more statistical power, and your own questions that you're asking, you'll be able to ask them better. Right? Another concern that came up was we don't have enough metadata for diversity analysis. And I think that's often true, but then if you're starting from zero, right, we know so little about human diversity that anything you can learn from your data is better than zero. Right? For example, even if you know nothing else, you know the age and sex of the donors, almost always, right? And that itself is a major axis of human diversity that we'd like to integratively analyze across many cohorts, many tissues. Then geography, you don't precisely know where they live, but you know roughly where they live, right? Because you know where you collected the sample, and you can guess to some approximation that they live nearby. In most cases, it'll be true, right? Or in many cases, it'll be true. Then as far as ancestry goes, there's people expressed, kind of some criticism about the very crude bins of Asian, white, black, etc., the self-reported categories. And we've discussed that already. But what I'd like to clarify is that from the single-cell RNA-seq data, you can algorithmically infer genotypes and infer ancestry in a data-driven way. So even if you feel you don't have that information, the information is there in the single-cell RNA-seq reads. There are, of course, ethics issues. Some of the people may not be able to share their FASTQ files to other data analysts to infer ancestry, in which case maybe we can pass you the software to run it on your own data privately, infer ancestry, and then just give that back as metadata, right? And then you don't have to share FASTQs at all. You can just share gene cell matrices. So all of this takes work, obviously, and it's not like now there's already a published paper with a gold standard, widely accepted method for inferring ancestry from single-cell RNA-seq data, but there's lots of teams putting in that effort. And those pipelines will be available, I think, faster than your data sets will be wrangled and shared in many cases. So eminently doable. Right? And even if you think you don't have a diverse cohort, you have age and sex diversity at least, right? Even if you feel you don't have enough metadata, in most cases you do. So it's a matter of the time and effort, and I really hope that, and we've already identified in all these sessions some promising data sets that we can add to the genetic diversity network effort. For example, Hari has a breast tissue data set that's quite diverse, already been generated. Kidney has a diverse data set. Nasopharyngeal, Jose's data set is expanding, coming on board. Immune is, of course, the centerpiece because so many people, as we discussed, have diverse immune data. Adipose is just getting started, may not happen in time for version 1 of the atlas. But still, we have enough to chew on, for sure. And so please email us, please contact us. Now that maybe I've hopefully reassured some of you that you have what it takes to learn something about human diversity. And yeah, that's it. Let's get started.
So with that, we are reaching the end of the second day. If you remember yesterday, it was all about the great things you could do with single cell. Today, it was really the hard problems, and this was really a working meeting today. Thank you for contributing to the breakout sessions, tackling these hard problems and making plans, making roadmaps. Tomorrow, we will look into the future. Ultimately, we want to have a positive benefit for medicine. We want to cure diseases. We want to make all of this information useful for humankind, and that will be the overarching theme for the morning session. In the afternoon, we will give you an update of the 6 EU pilot projects that the European Union has been funding, and then we will close with a very exciting session on future perspectives. So please come again tomorrow 9 o'clock in the morning, and you will not regret it. And even more so, come now for the dinner, because that you're going to definitely regret if you don't come and you see all those pictures on Twitter of us having barbecue outside in a very nice location. So this is like a 19-minute walk from here, just enough to kind of digest all the food you ate today and be hungry again. This is in the, so if you've got, so we will try to make all of you find the way, but if you get lost, ask for the Museum of Applied Arts, because this is the restaurant of the Museum of the Applied Arts. Join us there. It will be outdoors. The weather is very nice, and especially those people who might still watch us online. If you're in Vienna, you can join the dinner. If you're a virtual attendee and you're in Vienna, send me a quick email, because I need to have an overview of who's actually physically there. I need your name. But if you're online, you're visiting now, you're kind of watching now, and you want to join the dinner and see kind of all of the people in person that you've seen in all of these sessions, please feel free to join us to tonight's dinner. Good. With that, I would like to conclude and see you there at the dinner. Thank you.